All right, let's go ahead and get started. Welcome everybody to the first KBase webinar of 2021. I am Benjamin Allen. I am a researcher based out of Oak Ridge National Laboratory, and I've been on the KBase project since 2014. I'm going to be doing this intro to KBase today to all of you. So maybe some of you are brand new KBase users. Maybe some of you have used KBase in the past and are coming back to see what's changed or what's new. And maybe some of you are current users who are just learning to look more about the system. So hopefully we'll have something for everybody in here today. Presenting alongside me today is Dakota Blair. He is an engineer based out of Brookhaven National Laboratory. And so he has is going to walk us through our brand new narratives manager interface that he helped build. So yes, some exciting new features that we are debuting today as well with the new narratives manager. So we'll get started here. I'm going to stop my video and just let you all focus on the screen share. Hopefully you can all see this well. In the email that we sent to everybody about the webinar, you should have seen a link to this questions and answers document. This allows us to take your questions while while I'm presenting, and so we don't have to break up the time, and we have staff on the call who are going to, and in this document, who will answer any questions you have. You can see someone already asks for some guidance around uploading their reads, so you can just write your questions in there. And you'll see some links to different helpful resources, some of the links that I'll talk about today, to various sections of our website, to documentation, etc. So there's quite a lot here, so you should be able to figure out find what you're looking for with one of these links. All right, so I just want to start off with the kbase.us website. So if you're going to KBase, you got to go through kbase.us. This is our brand new, basically redesigned homepage. We just launched this at the end of last year, so we're still building it out, but there's quite a lot of useful information here. I won't go through all of it, but just a few highlights. On the homepage, you'll of course just see some basic information about the system. The research page is a really helpful place to sort of see what people do in KBase and what the various participants in the KBase community are up to. So we post things like narratives or publications that are we want to highlight here. We try to keep a running list of all of the publications using KBase there as well. The Learn section is going to have information about various analytical workflows in KBase, just different kinds of resources like tutorial narratives, etc., for you to learn how to use the system. Engage tells us, is maybe one way you could find your way here, because we show our events and training events or upcoming events, and as well as previous events with recordings of webinars. You can find all of that on the Engage page. We also have some different organizations or KBase organizations, which we'll talk a little bit more about. You can find some highlighted ones on here. And news, of course, is a great place to figure out what is going on with the system, any changes, any upcoming outages or migrations, things like that. You'll see all that on the news page. And the support page, eventually, if you're using KBase, you'll run into issue where you might need some support. So you can go to here to find links to our help board or just other information. The first thing you'll want to do is to sign up for an account if you haven't already. Um, this will probably just sign me directly into my KBase account since I'm already signed in. But if I was not signed in here, I could go to sign up, and this will show me some different options for creating a KBase account. You can create a KBase account if you have a Google account or you have an Orchid ID. You can just very straightforwardly create a account using one of those. Or if you have Globus, you can create a account using Globus as well. This is especially useful if you are a Department of Energy employee and you have secure data guidelines that you have to abide by. So Globus can be very useful for that. I will go ahead and sign in using my Globus account. And now I am in the first place that we come to in KBase. Oh, I might have just reloaded that. Is our new narratives manager. We're going to come back to this, and I'm going to let Dakota give a overview. But before we go into the narratives manager, I'm going to just go ahead and create a new narrative because in KBase, narratives are the primary working environment. The vast majority of the stuff you do in KBase, you do inside of the narratives. So, all right, so I've created a brand new narrative here. And what you can see is 4 different sections, basically. In the middle here is the narrative, as we call it. It's going to contain all of the app's data, workflow information, that we're putting together for our analysis is all going to be contained in this middle section. But right now, this is a brand new narrative, so it just has this welcome cell. And we can delete this cell if we don't want it. And you can see it's fully blank now. To the left, you'll see 2 sections. The top section is the data panel. The data panel, as you might suspect, allows you to manage the data you have in KBase. This narrative has no data yet, you can see. We have this big red add data button for you to add some data. So when you click that button, a slide out appears and it'll show you the different sections of different ways you can add data to your narrative. You'll see the first 2 sections, my data and shared with me. This is data that you have either uploaded and is associated with your account or generated on the system. And then your shared with me is going to show you any data that's contained in narratives that have been shared with you. And I've got quite a few, so it takes a little while to show, but you can see all of that there. Our public tab, I'm not going to click it right now because we're doing maintenance on it, and so it won't show you anything. But this is where we store our public reference data. Basically, we try to pull the RefSeq genomes at regular intervals from NCBI and use them as our public data backend data set, reference data set that we use for a lot of different applications on the system. So we pull that data and serve it up to all of you, and you can access that via the public data tab. You'll also find data from JGI there as well, like JGI phytosome or the mycocosm fungal genomes there as well. We have this example data section if you just want to play around with the various data types that might be useful to you. What will certainly be useful to almost all of you is this importing tab. And this is where you can drag and drop data into the system to import it. So if you have files on your computer, you can just drag and drop them into here. There are ways you can do a much, for batch upload. This is one of the reasons you might want to create a Globus account, is that you can use Globus to batch import, large numbers of files. So you can see I've just 6 minutes ago imported this file into here. This is a reads file. So what I'm going to do is show you how to import it. You can see some just interesting things about it. I can click this little drop down. I can see information about the reads. Look at the first 10, or well, it's not a text file, it's a zip file, but if it was a text file, it would show the first 10 lines. But so you can check them if you have certain files. But this is some FASTQ reads that are zipped up here. And I am going to upload them by clicking this upload button. So just before I do that, these other two buttons, this is the download button. So if you want to download something you have previously uploaded, you can click that and that will download it to your computer. Or if you want to delete something, you can of course click the red trash can button. But I want to upload this. And you can see I now have a importer here. So we have basically two stages to our import process. There's putting the file into this import section, which is called the staging area. And so this staging area allows you to transfer the data to your various narratives. So you first put it in the staging area, and now I will try to pull it into this narrative. So this is a single file. Someone asked in the question and answer, what do you do if you have a forward and reverse file? Basically, you import both of those into the importer. You'll drag and drop both of those into the importer, and then when you come to this stage, you will select the forward and then the reverse read, and you can set information about the sequencing technology. So this shows you the kinds of reads that we can handle via this importer. In this instance, it's a single genome. If you are dealing with metagenome reads, you would uncheck that, but this is a single genome. And this file, as we're uploading a single file, it is an interleaved file. So this is a very important box, and I'll say, we often get questions in the help desk about, people will try to, import a single file, but they don't check the interleaved box, and that will give them an error. So yeah, that's a very important box. So this means that this file contains both the forward and the reverse reads set. So this is all ready to go. And what we're looking at here is, it is this importer, but this is what we call an app cell in KBase. So basically, we'll cover apps in more detail in just a second, but this cell, as you can see, is a graphical user interface based way of showing how to run this tool. So what we're going to do is click this big green run button, and that will start the importer job for importing these reads. So you see I'm waiting, I'm in queue. Some things I can do in this app cell while it is running. I can check the job status, for instance, and you'll see now you'll see a sort of terminal based job log of what exactly the app is doing. So that's importing data, and as you can see here, the next section is that we want to run apps on our data. So we have this apps section here where we can select various apps that we might want to run. So this reads set will finish importing here shortly. I'm going to add some other data. Let's see, I've got all sorts of stuff here. I want to find a genome, so I'm going to go to this types filter here, go to genome. I've got quite a few genomes, and it will load the various genomes that I have. Let's go with trout bog. That's fun. So we'll get this. This is actually a meta genome assembled genome. Um, but what I can do here is I clicked this 3 dots here, and I can see all sorts of information about handling this data. I see some options here for I can delete the data, I can rename it, etc. An interesting one though is this show apps with this as input, and when I click that, it creates a filter here in the app section to show me things with this that will take this data type as input. So it filters our list, and I can see I have these various categories. Let's go with comparative genomics. And I see all sorts of various apps that I can use on this data type. So let's just go to a common one, which is insert genome into species tree. You can see it creates a new cell here for this app in the narrative. And I can set the various parameters, set the input objects, set the output names, and basically fill out what is needed to run this tool. So I click the plus button here, and I can select the genome, and I can set some parameters. So maybe I don't care about seeing all 20, but I want to see 10. I don't want to copy the public genomes to my workspace, but I can give some very, very generic names to the output here. And again, click run. And this app will start to run on the data that I put into it. And again, I can check the status as it, once it starts to execute at least. And there it goes. So now in just 10 minutes here, I've basically shown you the majority of what you do in KBase, right? You're finding data, you're running tools on it, you are and you're doing it all inside of this narrative interface, and not on command line or in your terminal, or you're trying to handle various Python packages and things like that. We have contained have built the container for these various apps and data to operate together in this environment. So all these things are running right now, and so we'll come back to them. But just to show you the last part of the narrative interface here, the top section, you'll see the toolbar. And so I'll just go from left to right. You see the hamburger menu. If I want to go back or see some information about what's going on, I can use that. I want to give my narrative a name here, and just call it intro webinar. And then I'll click OK. And it renames and then saves the narrative. It shows that little checkpoint dialog. I can see information about who created this narrative. Soon we'll be adding some more stuff like showing who it's shared with and so forth and so on. But right now it just shows who created this narrative. You'll see some a help button if you want to figure out what's going on inside of the narrative, or if you want to drop us a line about a question you might have. The kernel section, occasionally, you know, maybe your internet drops out or you get disconnected from the narrative. It will show a red dialog here that says disconnected. You can just use these top level functions to restart, or if you are, you know, interested to see anything about the Python libraries that this is built on, you can find out information about that. Importantly, here is the share button, and this allows us to share the narrative with other KBase users or publish a static narrative. So more often than not, you're going to want, you know, you may have a lab full of people that are all working on the same data, or collaborators across the country, whatever it is, you can find KBase users here to share your narrative with. So here's one of my colleagues, Zach Crockett. So I want to share with him this narrative, and I can give him various levels of access to that. I can provide view-only access, which means that he can see the contents of the narrative, but he can't change anything about it. He can't add data or run apps or anything like that. Or I can do edit and save here, and this means that Zach can add data, add apps, run apps, things like that, and save it. So he can, you know, basically do anything I can do except share. And then there's the highest level permission, edit, save, and share, which means he can also share with anybody. So this just gives you different levels of you know, privacy for your sharing. So I'll just give him view only. And if I ever want to take him off, I can click that red button. I can also associate this with a organization. So we'll talk about that a little bit later, where we have organizations in KBase where, you know, you have common access to narratives and data. Or I can share this narrative with the world and make it public. So at the top, you'll see this private section here. So if I want to make it public, I just click make public there, and now it is unlocked and it's public. So what this means, if it's public, is that anyone who has a KBase account can come and see this narrative. And when Dakota presents, he'll show a little bit about, you know, where you see public narratives inside of KBase. But I don't really want to make this public, so I'm just going to make it private again. This next, well, let me just say the save button is very important. KBase will automatically save as you run an app, or basically anytime you create an app, sell or run an app or do something, it tries to autosave, but just, you know, just like any other program, you know, you're encouraged to save your work whenever you are at a stopping point, or especially whenever you're shutting down your narrative. So the last thing I want to mention is static narratives, and static narratives are a relatively new feature for KBase, you know, just maybe 3 or 4 months old, that allows you to create a HTML version of a narrative that is indexable. It's you know, searchable, findable on the web, and I'll just show an example of this. This is from one of our developers, third-party developers, who has built some viral tools inside of KBase. This static narrative is available for anyone on the web to see, and you can just see it's got the you know, commentary section. You can see the various apps they've used. And yeah, you can just basically see everything that is inside of their narrative, and you don't even have to have a KBase account to access this. So that's a very powerful tool for really getting your work inside of KBase out there, and, you know, sort of bringing people to your data and your analysis, you know, as you did it on system. So this is a very very useful tool. So you can do that in the manage static narrative section. You'll see it's not public. If I make it public, I guess I should have kept it public. You can see I can make a static narrative for this. No static version exists yet, but if I click this button, it will create a link to a static narrative that anyone can see. And the interesting thing about this too is that the static narrative is a specific save state. So for instance, you might have a specific version of a narrative with specific apps that you use, specific versions of those apps, and you want to share that with the world, but maybe you want to continue to tinker in your actual narrative after that. You could do that and your static narrative will stay the same, but your working narrative would change. So that's kind of a powerful feature. Yeah, you can see here that this app finished successfully. So I'm going to go to the result tab here just to see what it says, and there it goes. It generated this nice phylogenetic tree for me here with the 10 nearest neighbors to this trout bog mag that I put in here. So basically, with various apps, you're going to have different kinds of reports. You know, the uploader, the assemblers, the annotation tool are all, and these comparative tools are all going to have different kinds of reports or tables that are built with all the results. There are a couple of ways you can download the results. Like, for instance, you'll see it's showing me this report in the cell, but if I want to view it in a new window, it opens this up and I can save the image that way. Or I can go to the bottom here where I have all of these links and files that I can download off the system. So you see a PDF, a PNG image file, these NUIC files that you can then take and use in something like RexML or other tree building applications. To do more customization on that tree. But you see, really, the other thing that happened when this app finished is that it populated data over here in my data panel. It says, 8 minutes ago, this tree and this genome set were generated. If I want to download that, I can click the 3 buttons here and go to the export download data. And you'll see I get some different options. I can export it as a NUIC file. This JSON is just a KBase specific file that shows how our data is represented on the system, our data objects, as we call them. For different types of files, like this genome, for instance, has some various types of things you can export as GenBank, GFF plus FASTA file, or I can put it back in my staging area. So that can be very useful as well. Seeing some people raising hands in the Zoom, if you will, please ask any questions you have in the Q&A document here. And yeah, seeing some folks are getting their questions answered. So thanks a lot for KBase staff doing that. Okay, I think I have covered the basics of the narrative. Oh, no, two more final things to show. So I've showed your data importer. It's still importing this data. It might take a little while. So we have our data, we have our apps. You can also add Markdown to the narratives, right? So you can see I can just write stuff. So this is all based on what's called Markdown language, or MD. So if you we've got some examples of using Markdown that are in our documentation page, but yeah, so you can just basically write whatever text, put graphics, as I showed in this static narrative, this is an example of somebody really using this to the max, and providing links and graphics and things like that. So you can put whatever you want there using this button at the bottom, add markdown cell. If you are a experienced bioinformatician and you are interested in running Python scripts, you can do that with the code cell. So KBase, this narrative interface, is built using the Jupyter notebook. So basically you can run scripts in these cells, and run multiple scripts inside the narrative, and you can run it against the data you have, or yeah. So we don't have much documentation about using the code cell right now. We will hopefully host a webinar later this year where some of our staff experts will go into that more. But yes, between all of these features, markdown cells, data, apps, you get the idea that you can use the narrative as basically an active notebook, lab notebook for your bioinformatics analysis, and contain all of your material inside of each narrative. So with that, I'm gonna hand it back to, hand it for the first time over to Dakota. So I'll stop sharing my screen here. And if you want to start sharing, Dakota.
Sure. Okay. So what I'm going to be showing you is the narrative view here, where we can see information about all of our narratives. And a little bit about me. My name is Dakota, and I've been working with KBase since about April of 2020. So I'm relatively new to the project, but I'm real excited to work with them. I got my PhD in mathematics in 2015, so I don't know as much of the biology as some of the other people on KBase. But despite that, I can still, as Ben was mentioning, I helped get this out the door. A lot of people had worked on this view, and I helped at the very end, and now it's here. So I'm going to show you some of its features. Okay. The first one I want to show is just that this lists some narratives. Okay, so right now I'm on my narratives, and so all of my narratives are listed here. And what you can do over here is you can select each different narrative, and it will change the preview over on the right side. And in the preview, I want to show you a few things. So wrangling Zoom right now. Okay, so in the preview, we have the name, the title of the narrative, what version it is, who the author is, which on my narratives is always going to be me, but we'll see in a minute that it could have different values. When it was created, when it was saved, what is visibility, who it's shared with, and also some information, some aggregate information about data objects. So like what kinds of objects are in there and how many there are. And then similarly with cells. So how many cells there are, what kinds of cells. And below, speaking of data, we can actually see each of the data items in this narrative. And that's what this right here is. And we can also preview the narrative itself. So when I click on that preview, I can see just all of the different cells in the narrative at the beginning, at the top of the narrative. Eventually, if you have a long enough narrative, you'll see there are more cells to come, and you'll have to view the full narrative to see the rest. But you can click either this link or the link at the bottom to open the narrative. And you also have a chance to do a few other maintenance type tasks. So for instance, if I want to manage my sharing or copy this narrative or rename it or link to an organization or even delete it, I can do all of that from this view, which I understand is, it's that you can't do that with the other one. The other one, you'd have to open the narrative and then do those sorts of changes, but here it can all be done very easily. So we've talked about these tabs, the data and preview tab, but let me talk about these other larger tabs here. These are filters for different narratives that you have access to. So my narratives are the narratives that I own, that I can do with as I please. Shared with me are narratives that have been shared with me that I can, use up to my permissions that were given to me whenever they were shared with me. Tutorials are tutorial narratives that just show you how to use the system. So for instance, here is a nice tutorial on genome analysis tools and features. So a lot of you who are asking these questions in the chat or in the document, you can look through this tutorial and maybe some of your questions will be answered there. And you can see we've done a lot of work on this one, so that's good. And then there is the public narratives, and so these are all the narratives that anyone has just made public, and which is really nice. And I'm actually going to show off a few different features here, looking for the narrative that Ben was sharing with us just a moment ago, because I saw the webinar where that narrative came from, and I was interested in seeing, okay, what does this narrative look like? How can I run that? And I remember that it was about viruses. So I'm going to look for viral. Yeah, and so here's the one that I was looking for. So this is my viral analysis end to end. And you can see that this is that same narrative. And I could open it, and you would see all those pictures. Well, I might as well do that. So I'm going to go ahead and open this. And so you can see that this is the same narrative that Ben was looking at earlier. And it's very simple to open these narratives and go through and see exactly what has been done. And they open by default in a new tab, so you can go back very easily and continue your search. But the other good thing about this is that this search here, it's not just searching through the titles of the narratives. For instance, this narrative also uses virus sorter. And so maybe I want to look at other narratives which include virus sorter, which, just so I can see what sort of, how it is used. So I can search for virus sorter. And this narrative comes up again, but also these other narratives. And so here's a narrative that I have very little familiarity with, other than it's in this list that I just searched for. But I can see that it also uses virus sorter, which is really nice. And so this search doesn't just search through the titles; it also searches through the data items and also in the preview. And then the last thing I really want to show is how we can sort by various criteria. So recently updated is the default, and you've got least recently updated and recently created, or oldest. So you can see what I was working on when I first began working at KBase, or like what the oldest public narratives are and what they have to do with. So that's pretty nice. And then also lexicographic. So if it's easier just to see them in a kind of alphabetical order. Great. And so I'll hand it back to Ben now. He already demonstrated using the new narrative button, which I thought was really nice. If this, let me go back to my narratives. This is here in case you're, you're feeling like there's something that's not loading right. And if, instead of reloading the entire page, you can click this, and it'll reset a few things so that you can get the latest data for sure, no matter what you have. Great. And back to you, Ben.
Awesome. Let me hit share
Want me to
screen.
stop sharing?
Yes, please. Thank you. And yeah, so we know we're covering a lot here, and just wanted to before I go into more, just call back to two valuable resources that you'll certainly find useful as you continue to use KBase. One is the help board. So I'm just on this support section of kbase.us. And if you click this go to help board button, you will, well, you'll probably see something a little different, but basically this public help board allows us to gather any tickets that you might have, support tickets. You can see here some different ones, errors that people have encountered, or just questions like, how do I delete my narratives, right? So Miriam Land, who is a KBase staff member based out of Oak Ridge, is one of our, or is, I should say, the top help desk problem solver. She will definitely help you solve your problem, or at least figure out what the problem is on our end. And I think she's helping solve some of your problems and answer some of your questions in the Q&A document right now. So yes, please, if you ever run into any kind of issue, you can create a ticket on the help desk, and we will do our best to address that. The other thing I just wanted to mention, and there's links to this in the Q&A document, there's links to it on our website, is our docs, our documentation website, docs.kbase.us. So this is another relatively new resource that we've been putting together, and it just really covers a lot of different, everything we've covered today, in addition to, stuff like doing common workflows. Someone was asking about de novo assembly in the chat. You can see lots of different resources on the documentation website. So Ellen Dow and Zach Crockett and myself and others, have tried to capture as much as we can here. So even the questions that you all are asking in the Q&A doc today, we'll try to add some of this to the FAQs on this documentation site. So just two more resources to help you learn how to use the system. Okay, let me go back to KBase here. So yeah, thank you, Dakota, for all the work on this new narratives manager and for showing it to everybody. This is very exciting. As I said, I've been on KBase for a long time, and this is really, really exciting to have this new manager where we can do lots of useful stuff without having to go into the individual narratives. So continuing to walk down this toolbar on the left-hand side. So we mentioned organizations. So orgs in KBase are a feature that, basically, say you have your lab, say you have a specific project where you might be trying to pull together many different users and researchers from into some common environment. Orgs allows you to do that, and basically within an org. So I'm just going to open one up here. This is for the Enigma project. So it will load the organization. This allows you to, as I showed when I was sharing my narrative, if you're just sharing a narrative with one or two people, it's very straightforward. You can just type in their username or search them. But if you're trying to share it with hundreds of people, it starts to get challenging. So an organization allows you to have members. So various KBase users become members of the organization. They can request to join an organization if they're not a part of it. So not everyone can just come and go. You can add users manually, or they can request to join. But it allows you to sort all of your organization's narratives and data and give access to the various parties they're in. So here I have a list of narratives that are associated with this organization. You can see they're working on various, genome drafts and stuff like that. Some of these are able to be seen by everybody in the group. Some, have, there's permissions within the group as well. So, you can divide up the ability to, give access to those things. You can associate a new narrative with a group. So if I wanted to add a narrative, I could click. I won't add it here, but because it's not really my group. But if I wanted to add a narrative here, I could select one, and oops, it took me all the way back. Just wanted to show some more stuff that's included here. So, I can also have, description, right? This is a very nice description with the lab group picture and all of that kind of information. So, maybe you're using your organization to, kind of broadcast your research and data to other KBase users. Maybe you're just using it to, organize the work of your lab group, but it gives you a lot of flexibility in terms of how you want to deploy it. But would encourage you all, especially if you have colleagues at your institution or in your lab that are using KBase together, to try organizations out, see if see how it can be useful for sharing the data and narratives that you all are working on together. I just will show you some other public orgs that are useful. So the KBase educators org, there's a group of people that are using KBase to train their students on bioinformatics. So you'll see a lot of different narratives in here that show different exercises and different kinds of material for teaching students how, not just how to use KBase, but how to do some of these fundamental processes for bioinformatics. So a lot of different stuff in the educators org. Very, very, very valuable resource. So go and check that one out. And yeah, there's other ones too, like for instance, published genome scale metabolic models is where Chris Henry and others are trying to curate a set of genome scale metabolic models that are very valuable to the community. And so on. There's just a lot of, we have over 110 different orgs right now. You can filter the orgs or show by, sort by different different things like org name. So yeah, just check out orgs and let us know if there's features that you would like to see within orgs that would make it even more useful. I should say, to create an organization, you do that by clicking this blue Create Orgs button, and that will give you the various dialogues, name and URLs and stuff. This is just all of the descriptive text for creating your org. So I will keep moving down the left-hand side here to catalog. So the app catalog, a long time ago, KBase just had a few apps, so it was very easy to show, doing QC, assembly, and annotation. Now we have quite a large number of apps. I think we're over 300 at this point. But so we have a very large catalog of apps. So the power of KBase is its ability to host a lot of open source third-party apps in this kind of common environment where the data is interoperable between the various apps. So, different types of reads can go through, the same assemblers, and the outputs of those assemblers can be used in the annotation tools and so on. And, things are able to work together. But sometimes, making choices about, well, which app should I use, or, is this the right tool for what I'm doing? That can be challenging. So the app catalog is meant to give you a couple of ways to find the tool that you need and to see if it is indeed the right tool. So you can see here a variety of categories like read processing, genome assembly, and then under each one of these categories, you just see the list of apps. So very popular app here, Check-In. I can tell it's a popular app because of two pieces of data, metadata affiliated with it. One is the favorites. So it seems that 56 people have made Check-In a favorite app of theirs, and I will add myself to that list. And now I'm number 57. And next to that, I can see that Check-In has been ran, over 21,000 times by various KBase users. So that's a pretty popular app when you can kind of see many things are run, maybe a few thousand times, maybe a few hundred times. And I can hover my mouse over and see a short description, but let's say I want to see even more information. I just want to click that and it will open up the app page with all of the description of what this tool is, how it works, and how I might use it. So you can just see a lot of description here. So this is a very high quality app page, I'll say, is because it has a lot of information that it tells me, what I'm looking at in the output report, how I might set the parameters, etc. And I just keep scrolling down to show what else we can see here. Some information about, basically the KBase data types that it takes, even more information about the parameters. I can also see what publications are related to this app, and that's very important because, sometimes we see people, using KBase, and they'll say, well, I did this stuff in KBase, but they don't really cite what they did in KBase. So it's very important that you not just cite your use of KBase, but you cite the use of the specific tools that you're going into your publication. And you can even go out to the location of this tool on GitHub if you want to, investigate the code or anything like that. And you can also do that, see if there's a suite of tools associated with this app. So there's two apps that are using the same GitHub repo here to build both of these apps. So I can go out here and see what's going on, if there's changes, if is someone working on something. You can peek behind the curtain a little bit if you are interested in that. So I'll go back to the catalog and I can do some filtering, do some searching. Maybe I'm looking for an assembler, so I just type in assembly, and well, yeah, I can see a symbol with no no vowels is probably the best search term here. So I can just see the various assemblers that I'm might be interested in. KBase does, we'll do a webinar later about our software development kit. But if you are a computer engineer or scientist and you have your own tools that you want to add to KBase, you'll see this add module link here. It's not quite as easy as just pushing the button, but this does allow bioinformatics software developers to add their tools into the KBase app catalog, use our compute power and data backend and all of that to power their tools. So we're seeing more and more people do just that and add their tools into KBase, which is very exciting. Keep going down the left-hand side here. So search is a tool that allows you to search for data and narratives in KBase. So I will just type in the name of a genus here and hit search. And what happens is I see a combination of public, like shared user data, not necessarily private user data, and the reference data that is matching the hits or is generating hits for this search term. So you can see in reference data, it found almost 2,000 hits for this, and 125 of that was various public genomes. And so I can scroll down, and it's quite a lot of information in this view, but maybe I want to view less detail. I just want to see a list, so I can use this filter to pare it down. And now I can just see a whole list of these organisms. Maybe some of these were the ones that I'm looking for. So let's say I want to add them to a narrative. I'll just check these boxes here. And this blue copy selected button appears at the top where I can click it. And I see the objects I've selected, and I can either copy them into a new narrative, give that narrative a name, or I can just put them in an existing narrative. So that's two ways I can move this data around relatively quickly. So this view is a little easier to find and add data. If you're looking for public reference data, than using the public search browser, which, as I said, I won't show because it's under maintenance right now. But there's a little bit of redundancy in the system, which is good. So if the public search public tab is down, you can always go to the search tab here and still access the data you're looking for. This feature is, as the big beta sign says at the top, is something that we're still working on. But y'all are the users. So if you have ideas about what kinds of features you want to be able to search for, or how you would like to use search to enhance your use of the system, please go to the help board and make a ticket and let us know how you feel about search. I think that should pretty much cover search. It does find narratives too, so you can see these public narratives here. It's finding some stuff that I've made. It has this in here to indicate that it is a narrative, and there's things inside this narrative that may have hits. So that can be useful if you're trying to find public narratives that are interesting to you. So keep working down here to the jobs tab, and it is loading that up. Maybe it is not loading that up right now. I think I recall seeing some chatter that the jobs browser might not be operating at 100% right now, but no. Okay, here we go. Yes, now that loaded as it was supposed to. So you can see my jobs here. Not too much going on in the previous week for me, but you can go back through all time, for instance, and see all of the jobs you've ever ran on KBase. Basically, this if you're running a lot of jobs and you're as you start to build more and more narratives, and you might have multiple narratives where you're running several jobs per narrative, it can start to get a little challenging to remember which tab you had open and stuff like that. The jobs browser just allows you to see this from a top level view, where I can see the narrative I ran a job in, the app I was using, when did I submit it, how long was it queued, how long did it run, etc. Which server did it run on, did it complete, did it have an error, stuff like that. So that's very handy if you're managing a large number of jobs and you're trying to remember which narrative that was running in. So I can see even to the job log level what happened throughout a whole job run. So This can be very useful, especially when you're running into an error, going and looking back at the job log and seeing, what happened. Like, so this one generated an error. So I will go and see what happened a few months back. Right, so it didn't find any hits, so it generated an error here. But anyway, if you're trying to see what exactly is happening at the fine, line by line detail, you can do that in the job log, and that can be helpful for understanding how some of these apps work. Just to be honest, right? Like it will, depending on how much thought the original developer put into sectioning the various runs and labeling what's going on at each step, you can kind of see how these things iterate through their algorithms and processes. So so I can see the log, or I can just see information about the job ID. So this is useful, right? Sometimes when we're trying to track down a specific issue, we'll find the job ID here. So providing that kind of information in your help desk ticket is a great way to make it go a lot faster. So I can do more filters here if I want to do a specific app down to that level of specificity. So that can be really useful for people when you've hit the level of thousands of jobs, to find specific instances. So yeah, jobs panel, very, very powerful. Keep moving to account here. So when you first create your account, you put in some very basic information. And this does is help you, remember what your username, when did you create your account, this kind of stuff. If you So we often have people that will make multiple KBase accounts for one reason or the other. Maybe they forgot their login. Maybe they switched to using Google or Orchid ID. You can link multiple accounts to each other. So I don't have anything linked to this one, but if I have multiple user names for whatever reason, I can link them. And that just what that allows you to do is access narratives and data that were affiliated with your older account. So yeah, if you sometimes we have users that they used KBase 2 years ago, they can't remember how they logged in. Then we eventually will find their account and they link it to their new account. So that's a very common thing that happens. You can track sign-ins, and if you have security concerns for your data, you can always check things like sign-ins and where people were signing in to access it. It's not here, but it's related, is the profile. So you have your account and then you have your profile. So you can access that via this graphic at the top, this avatar at the top right, and load my profile here. So there's me and my dog Cookie. And I've got just stuff, like information about research interests. It will load my organizations. This is, another part of the system that I would say, we're working on and are looking to build out more features that connect to it. So, we appreciate as much information as you can provide in your profile section, just because it does help us, understand, who's using our system and what they're interested in. But, eventually we would like to use this stuff as the basis for some more social kind of features. So you can help us do that by filling out your profile. And so you can click edit profile here to do that. Similarly to the social features we're trying to build, the last section here is feeds. So feeds tells me what's going on in various organizations, or if somebody requests to join an organization, stuff like that, you'll find all of that in feeds. This is definitely one that you all can help us make more useful by giving some feedback on it. For instance, you can see here, I've got 8 notifications to my account and it's mostly people looking to join this educators group. I think they have all been added, so I will mark as seen, and that clears the notification. Yeah, so feeds basically just gives you some indication of what's happening in your orgs. That's the primary function of it right now, and it also you can see information about system outages and stuff like that. So as you can see, the RefSeq public genome database is being worked on right now. So, okay, we're at near the end here. I just want to just show 2 examples I think that are very useful illustration of like how people are using KBase in practice. Some of you maybe were hoping for a little bit more like nitty gritty KBase analysis, diving deep into stuff like assembly and annotation. We will do webinars on that stuff in the future. We've got some recordings of webinars in the past that we've done on those type of analysis workflow type tutorials. But so we didn't do much of that today, but hopefully you got a greater sense of what KBase is, the various parts of the system, and the features. But I just wanted to show some examples of some how our users are putting the system to use. We get a lot of people making genome announcements in KBase. So this is a recent genome announcement that came out last year, where they were able to isolate this tough to isolate microbe, rosin microbiome out here at Oak Ridge. And you can see all this information about what they did to isolate it, but here you can see in the paper they have dropped a link to their narrative. So I can open that link, and I've actually already, oh, nope, sorry. So it will load up that narrative here, and I can see, just as they did on system, this example of genome annotation and assembly and annotation. So if I want to understand what they did to build this genome, or if I just want to like download the data and like I want to, I don't want to wait, right? I want to see this genome, and I want to load it into my own narratives and start running analysis on it. I can jump straight from the paper that I'm reading right into their narrative, where all of this data is stored. You can see here, I'm in view only mode here, so I can't really do anything to the content of the narrative, but I can see everything that they were able to do. So, I can go and check out their configuration of their assembler here, or sorry, this is an annotation tool. I can see how they've configured these various parameters, and I can just explore the genome as well. So here's the actual genome. I want to go and browse features, so it will build me a list of all of those features, and let's say I have something I want to search for. Suddenly I'm, taken into their lab notebook, right? And I can just start to see what's in there that might be interesting for me as a researcher who, might be interested in this organism. So this is a really powerful tool, and we encourage you to do this, to use public narratives, to use static narratives. Here's another example of a static narrative where this researcher is linking to a lot of different information and a lot of different analysis that they have done in KBase. So they're kind of like serving up the overview in this static narrative, and then allowing me to go and view the public narratives that they have generated, and again, all of the data, all of the analysis is contained here. I can make a copy of it if I want, or like I said, I can go and download these genomes. Well, when it's anyway, I don't know why that's struggling right now, but we're seeing users take these various features and make something useful for them out of it. And this is just like a creative way that we've seen users combining both static narratives and public narratives to share their science with the world. So a lot of exciting examples, more and more, seeing a lot more examples like this come every day. If we can help you and be of any service to any of y'all in promoting your research, showing you examples of using the system for various things, please just reach out to us. engage@kbase.us is the best place to do that. Especially if you're trying, if you're doing your experimental planning and you're trying to decide, what am I going to do with all of these reads that I'm generating? We can provide that some of that advice and consulting to help you make good decisions on the system. We can't really do the science for you, but we can at least kind of point you in the right direction about which tools or considerations you might have. So really appreciate everybody who stuck around to the end. We are going to share this recording as soon as we're done. I'm just checking the Q&A doc here to see if there's any unanswered questions that I can get to. Is KBase able to handle strand-specific RNA-seq data? That is a good question. So we do have, I would say it's certainly, we're certainly able to get RNA-seq data on the system and transcriptomic reads, and we have various tools to perform the assembly and mapping and things like that for transcriptome assembly and differential expression and gene enrichment and things like that. As far as that's something we'll have to put to some of the experts about what they might say about detection for antisense transcripts. But yes, we can do at least some of the basic transcriptome analysis if you're trying to look at things like transcripts per million and identify differential expression in data sets. So suppose you already assembled 5 genomes. How do you simultaneously run them to determine the clustering or placement of the genome in the same tree? Yeah, so this is a good question because it's this is some of the KBase specific kind of things. This data type there's 2 different tree builders. There's the insert genome into species tree, and then there is the insert set of genomes into species tree. So right, you can see both of these here. So the difference being that this one handles a single genome, but this one handles a set of genomes. And why that matters in KBase is that we have different types of data objects for individual versus sets of individual genomes. So you have the genome object, which is like this one, and then you have the genome set object, which is a set of genomes. So this is certainly something that we're thinking about all the time, like what's the best way that we can kind of organize data in a way that it's useful. And we're currently experimenting with some new ways to do that. But the way it works right now is that sometimes there are going to be just tools that are deal with the plural. So like this insert set of genomes where you're trying to do a kind of batch analysis. Different apps handle things differently at this point. Some apps will have it where like I'll just open up an annotation tool. Yeah, so another example, annotate domains in a genome versus annotate domains in a genome set. That's like that's one where we've sort of split it into two apps. But an example where you can. Oh, well, that was not a good example. Now, why am I failing to get a good example? Anyway, some apps you'll see have a plus sign where you can add multiple data objects on the front end, and some have you kind of select from a list. But anyway, that's long story short. There are different types of sets in the system. So assembly sets, genome sets. Read sets. And if you go to the utilities section of the apps panel here, you can see this is where a lot of the tools for combining different types of data into a set are located. So. And, again, the easy trick to finding the tools that will deal with the type of data you're going into it is to click these three buttons and click this first button on the left, show apps with this as input, and that will filter down to the tools that will take the genome set. So yeah, you'll notice just different apps have been built by different people, and just have different approaches to how they organize the input objects. Like this one, you can see it can take either an assembly or a genome. Other tools may be built where they've built one tool for the assembly and one tool for the genome. So we're always kind of trying to figure out how to optimize these things and prevent too much redundancy where it's unnecessary, but have redundancy where it's helpful. So hopefully that helps a little bit. How many developers are working on KBase? That is a good question. I think it's like 20 at this point. Not all of them are, and that's like people that are coders, DevOps, like yeah, I think that's not 20 full-time staff. That's much fewer than that are full-time staff. So a lot of people are on our staff are working between multiple projects. Some are full-time devs. I'm not exactly sure how many full-time devs. I think less than 10 of those 20. So we are actually a pretty small team. I think in total we're at like 45 team members across the board. So there's changeover that all the time, adding new people, and but yeah, we're a relatively small team. Kind of organized between sort of systems architecture, DevOps type people to the coders and front end devs like Dakota, and then there's those of us who I am not a developer. I am a biologist, and so I talk with the scientists and train people how to use KBase. And there's people in management and different types of roles. But I'd say we look like a small modern software company. That's sort of how we try to organize our project, is using those common principles that you see in a lot of kind of startup companies. So. Do you have bacterial genome indel calling apps who accept FASTA files? So we do. You can, yeah. So for if you wanted to look at, yeah. So we don't necessarily have. We are building some structural variance tools, and we're incorporating them into the system right now. You would find them in in the beta section. I didn't talk about that, but I'll show you really quickly. Yeah, so you can do some degree of like structural variance and GWAS type stuff. Yeah, so structural genomics or sequence analysis. You'll find some of this in here. But what I just did, more importantly, is switch to the beta section of apps. So in the app catalog or app panel here, you have release and beta. So our released apps are the ones that we feel meet the criteria for successful runs and general thorough development that we think are ready to share with you all. And our beta tools are the tools that are in development that we're trying to release and we're actively working on. So, feel free. We serve these beta apps up, so feel free to explore what's in there. No guarantee that it will work because it's in development, of course. But yeah, you can just see there's some new categories that are not currently in release and so on. Yeah, so check that out. But yeah, so we don't have anything that's released right now that I think that would be super useful for indel detection. But that's something we're working on. And I don't see anything else. So with that, I think we can shut it down here. But thanks again to everybody for sticking around. Hopefully you found this to be useful. And we encourage you to sign up for future webinars. We're trying to do at least one a month. And if you have ideas, right, a lot of you had questions about very specific apps or analysis workflows. Let us know what you want, what you would like us to cover on these webinars, and we'll make it happen. So thanks again, and looking forward to seeing y'all's research come out. So thanks, and good luck using this system, and let us know if we can help.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE_KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.